Hello again, everybody. So now we call the third speaker of the session, Alexander Helm from GSI, who is going to discuss immunological effects and give us details as anticipated by our first speaker.
So thank you for this opportunity to speak here. Can you also see my laser pointer?
Yes, very well.
Very nice. Okay, so now I'm going to tell something about immunological effects of radiation and with a focus, of course, on hadron therapy. So what you have seen so far is that radiotherapy is mostly meant to inactivate cells. You irradiate, you get your DNA damage, and the ultimate aim is cell death in terms of inactivating tumor cells, destroying the tumor, leading to shrinkage of the tumor. And you have also seen that due to the special physical features, heavy ions, mainly carbon ions, are of advantage here, and they are more efficient, more effective than photons, and that is what's used here, and they spare better the tissue. But however, there's more than this in radiation than simple cell activation. It can also be immunogenic. It can trigger immune effects that then not only work also on the primary irradiated tumor, but can also reduce tumors and metastases out of the radiation field. And this is acknowledged to be due to the immune system. And that leads to something called abscopal effects, which are very rare effects if you only have irradiation, but they have been observed in clinical cases too. Now, actually, especially with carbon ion therapy, the local control of a tumor can be very good. The problem comes that still with this therapy form, the overall survival is still then lags somewhat behind this local control. And this disappointing fact is mainly due to metastatic cancer disease, which accounts for most of the cancer deaths actually, and normally is something that you treat palliative with the goal to improve the quality of the life of the patient, because you would need some systemic treatment, along with the local therapy like radiation actually is. And this is where exactly it matches with the immune system. So the immune system actually protects us from cancer. That happens daily, that the immune system removes mutated cells. And this has been shown in experiments. For example, when mice are exposed to a carcinogen, you compare immunodeficient mice with immunocompetent mice, so mice that do have a functioning immune system. You will see that the mice with the deficiency develop much more cancers than with the mice without, with the functioning immune system. And this shows that the immune system has a really pivotal role in contributing to cancer defense. And that is shown also in populations, immunocompromised populations, like patients with HIV or transplant recipients that do have a higher risk of cancers as compared to the normal population. And that is due to the immune system actually reducing efficiently these immune cancer cells. So you have the cancer-developing cancer cells, mutated cancer cells that occur, and they are normally eliminated efficiently in the immunosurveillance progress of the immune system. But then they can also escape. This is shown in the next slide a little bit, somewhat more in detail. You do have the cancer cells. They are eliminated by a vast response of a lot of immune cells. They are eliminated, they die. This happens to the presentation of class 1 MHC. Those are molecules presenting antigens to the immune cells on the surface. You have the processing of antigens, and the tumor cells are recognized and eliminated. However, along in the genetic instability, the heterogeneity of the tumor, and the immune selection, so that the immune cell take out all the cells that it can take out, but eventually we'll have this one cell that can survive and has some strategies. We will see later more about that, how to evade it. It will do so and then can grow, and then you're in an equilibrium phase. And eventually you escape from this phase and the tumor can grow without the immune system being able to detect it. And even the tumor will be able to manipulate the immune system. We will see later a bit more on that. Just briefly some words on the immune system. So it has a lot of components, a lot of different kind of components, a lot of different kind of cells that are either innate or adaptive part of the immunity. The innate part is normally the one that responds faster. The adaptive part takes some more time, but it's then also more efficient and more specialized. And it has also kind of a memory function that is shown here with pathogens. So what happens with pathogens, you can kind of take that also given for cancer treatment, for cancer defense. So you do have also a memory function of this immune system. With respect to cancer, you have this so-called cancer immune cycle, a very famous picture that is known in this context. So you have dying tumor cells, they release antigens, they release DAMs. We will later see a bit more in detail what that is. All of that leads to immune cells, antigen uptaking cells, antigen presenting cells like dendritic cells to attract, to take up these antigens, getting activated, and then traveling to the next lymph node where they would activate CD8 T cells. These activated CD8 T cells will then clonally expand, and they will travel through the blood system, through the lymph system, again to the tumor site, and would recognize and inactivate tumor cells. So this is kind of a circle that is then also a feedback circular loop that will enhance also. Antigen-presenting cells, there are macrophages or also dendritic cells, as shown as an example as a dendritic cell that is attracted by some signals, will then take up the antigen, and then along with the course of the maturation and activation, will process the antigen and present it on its surface on these MHC class molecules. You have also some more receptors that are then presented. That happens along the change of this of the maturation, of the activation of the cells, and then they can present all of these signals, all of these molecules, the antigens, but also their receptors to other immune cells, which are also attracted by the release of cytokines. They take up these antigens by phagocytosis or pinocytosis or other processes. All of them share that everything is uptaken, is processed, and then parts of these proteins, very small parts, peptides, are actually presented on these very MHC molecules, presented to other immune cells, or also recognized on the tumor cells eventually. Now, when these cytotoxic T cells, the CD8+ T cells, are activated, they have several mechanisms. So first of all, they recognize with the T cell receptor and the MHC1, where the molecules on the cells are presented, they recognize the cells that are either infected or tumor cells, so they have something that is non-self. And then they have a couple of lytic or non-lytic mechanisms to fight that. The lytic mechanisms normally work with granzyme B and perforin, which opens the cell and leads to the lysis. They also can interact via the CD95 and its ligand to induct apoptosis, or non-lytic mechanisms which involve cytokines, which will attract further immune cells or NK cells, natural killer cells, to actually kill the cell. These processes, the interaction of the T cell with the other cells, tumor cells, macrophages, or dendritic cells, are not only regulated by one receptor, but of several receptors, some of which are activating and some of which can be inactivating. And so this is a balanced process, and it is very important not to overshoot. That is an important factor, but it can be hijacked by the cancer. And then this has been something that where actually the Nobel Prize was awarded in 2018 to these two scientists for having discovered this molecular axis, where you can actually interact with checkpoint inhibitors. Those are antibodies. It's now the most important part of immunotherapy, which is kind of a standard. It's for some cancers became a standard of care. And you can use these antibodies, the immunotherapy, to stop, to block these inhibitory signals, which can either be in the priming phase, so when the dendritic cell, the activated dendritic cell activates the T cell, or this can be done also in the effector phase, when the T cell would act on the cancer cell to induce the cell death, as we've seen it before, and then you can give checkpoint inhibitors to block this negative signal, and then you kind of boost the effect of the immune cells. You boost what the immune system actually already does. A problem of cancer is that the tumor microenvironment can actually alter this immune function. You've seen in the development that, of course, you have immune selection, and then you get cancer cells with different features that can hide. And there's a lot of processes going on that can lead to this. You can have loss of this MHC antigen on the surface, on the cell surface. This means the cells are simply not found anymore by the immune system. You have recruitment of regulatory T cells that are actually down-regulating the response, because everything is not only activated, but also down-regulated. You always have a balance, but the tumor shifts this balance towards something that is immune suppressive. And he has a lot of ways to do that. You have immune suppressive cytokines that can signal, again, a down-regulation of the immune response. Hypoxia is an important factor that also plays a role in that. It can lead to, it can suppress natural killer cells, can induce apoptosis, and then can recruit, again, more of immunosuppressive cells or tolerogenic cells. Or it can induce the polarization to the M2 phenotype of macrophages or also neutrophils. Those are actually related to tumor progression. So it can hijack the system and can basically hide from it, but can also manipulate it and use it for its own progression. They can also create immune suppressive environments by simply excluding immune cells or by recruiting cells that are immune suppressive, as we have seen. There are several ones: regulatory cells, MDSCs, myeloid-derived suppressor cells, or M2 macrophages, that by some certain signaling are more attracted than effector cells. And they can exclude them also along the growth. And it has been shown that this is also related to the outcome of the immune checkpoint inhibitor therapy, the therapy that we have seen before with the antibodies. So if you have a tumor that is infiltrated by immune cells, of course, you do have a higher chance that in these patients, in these cancers, you do have a response. Is the opposite is something where the tumor stroma is actually excluded from these cells, and then you do have also a low response. There are several more strategies by which cancers can evade from this immune surveillance. Some of them we have seen, others more in detail, and also are found basically in several patients. So there are a lot of strategies how the tumor can actually hide and evade, and that is also the reason why they do develop. Now, the good message is that radiotherapy can convert the tumor microenvironment in something that is, again, a bit more immunogenic. This happens with a kind of in situ vaccination. So you have a non-immunogenic environment before, and then when you irradiate, you have a lot of processes going on that would actually trigger, again, this cancer immune cycle, like you have seen it before, again, with the dendritic cell taking up antigens, getting activated, migrating to a lymph node, activating cytotoxic T lymphocytes, which would travel again to the tumor site and then interact with the tumor cells and make them actually die. And there are a lot of other molecules also in this process. So it can be kind of reverted by irradiation that is known. That actually sounds a bit too good to be true, and that is exactly the case. You do have immune stimulatory signals, that's true, but you have at the same time for radiation also immune suppressive signals. That's the two-phased nature of radiation. And you can either induce via some pathways, and you can attract cells that are related to tumor destruction, so the M1 polarization of tumor-associated neutrophils or macrophages. You have CD8 effector cells. But via some, especially other patients, especially TGF-beta, you can also attract cells that are related to tumor progression that are immunosuppressive, like myeloid-derived suppressor cells or the M2 polarized fraction of these cells. It's not exactly known what exactly which dose or what exactly in radiation triggers the one or the other. Classically, it has been assumed that low doses tend more for immune suppression, while higher doses go more for stimulatory signals, but this view has been challenged recently also. So it's not exactly clear in which way, which dose you have to address it, to use it in order to get the one or the other. It is not just important that the cell dies, but it's also important in which way the cell dies. The cell death is in the best case immunogenic, so that it can trigger these immune effects as we have seen it. And there are several signals, several things going on that is needed for an efficient cell death, for an immunogenic cell death. It's a whole thing of damage signaling, several ones that have been tested and known for that. And radiation therapy is actually known to feature most of these. So it has been shown for most of them. And these signals are very important along what we have already seen. The recruitment of dendritic cells, the activation of dendritic cells, antigen uptake, they may facilitate the uptake of the antigens, the maturation, and the attraction of further immune cells. So all of them play a role, and the more you have, the better it is actually for the therapy, to have a systemic response. The immunogenicity of radiation depends on two things. That's antigenicity and adjuvanticity. So to have a neoantigen induced by radiation, a mutation that is induced by radiation that can then be also seen as a de novo induced antigen on the cell surface, is simply not enough. So that's crucial, that's very important, it must be there, but it's not enough. You also need adjuvanticity. Those are the danger signals that we have already seen, that you need to attract the cells. And in radiotherapy, both play a role. Both are needed, and both are actually were found to happen, to take place. And you need your tumor neoantigens induced by radiation, but you also need the release of this danger signals along the immunogenic cell death. This is a bit shown more here, for example, in an example. So it's not enough that the dendritic cell takes up the antigen after the radiation, that when it's then shown on the surface. It is also important that you have your damage-associated molecular patterns that facilitate the uptake, or they simply activate the dendritic cell and lead to its maturation. Now, radiotherapy has been shown to be mutagenic. So what concerns the antigenicity, this has been nicely shown in this comprehensive study where they were comparing radiotherapy with other potential mutagens, and they could show that radiation was actually inducing mutations, was rather one of the very potential things to really induce mutations. So mutagenicity is nicely actually proven. It has been shown also in this paper, in this work, showing that radiation-induced neoantigens can also be used. They can be translated in functional CD8 in cytotoxic T-cell response, as has been shown here with the tetramer staining, where they used a cell line, irradiated it with 4-Cray, and then cultivated the cells for some weeks to see which stable mutations they have. They identified a couple of stable mutations in this cell line, and then they tested them also for the response on T cells. And what they found was that if they were taking T cells from these tumors of this cell line versus some irrelevant T cells, that is cells not from another tumor cell line, they could see that those very ones were actually reacting in the tetramer staining, showing that these immunogenic, so these mutations were immunogenic, they were there, and they could actually act in the mouse. That translated also into functionality of these T cells, as done here with an assay, with an ELISpot assay, showing that they would act on the tumor cells to inactivate them upon activation via these mutations. The question then is in antigenicity, especially whether this is improved with HITRIplus therapy, and there's rationale that this can be like that. We have a more clustered DNA damage. You have seen this for sure in the presentation by my colleague Walter before. You do have clustered lesions, they have a different pattern, and yield also a higher damage, unrepaired damage. And also the gene expression with respect to DNA repair can be different for higher Li particles. So the idea is that HITRIplus therapy can actually further improve the mutagenic landscape of tumors, especially of the tumors that are with a low mutational burden, which means actually more difficult to be found by the immune system. So high LED particles can also feature a different mutation signature that has been shown in p53 deficient cell line, and translated also in the mice that were deficient for this. And you can see that iron ions are actually more effective in inducing carcinogenesis, mutagenesis, as compared to photons. If you see the fraction of tumor-free mice. For chromosomal damage, it's also been found that the repair time, you check on the repair time, these damages, if you're comparing with photons after carbon ions or nickel ions with an even higher LET, the remaining damage is more persistent and there's a higher amount of repairing damage. So this actually speaks for antigenicity, an improved antigenicity with hadron ion therapy. I said already, adjuvanticity is as important as antigenicity. So just to remind you that you need all this kind of danger signaling that radiation can trigger. You need that along with the antigen uptake. And also the adjuvanticity, the danger signaling, has been shown to be increased after carbon ion radiation. So these are examples showing AGM-GP1 release or calreticulin exposure on the cell surface. And for AGM-GP1, these papers could show that carbon ions are more efficient as compared to photons, so significantly higher increase of the release of AGM-GP1 in the medium. And it has also been shown to be dependent on the LET. So this is a typical, I think we're comparing entrance channel. This is typical for the entrance channel versus a typical value, LET value for the spread out Bragg peak. And you see that also depends on the LET. And also for calreticulin, this is a paper showing that carbon ions are more efficient and faster inducing this calreticulin on the cell surface. And this is some data from us where we could show in an osteosarcoma model that carbon ions were more efficient. So it's also important in which way the cell dies. There are different types of cell death. There's not just only one type of cell death, there's several ones, some of which are being more immunogenic, some less. A very potent and immunogenic process is necroptosis. It's very immunogenic. And the more immunogenic is a cell death, better it is also for a systemic response. And there are some data showing also that carbon ions can be more immunogenic, that it can trigger a different type of cell death, which can be like necroptosis being more immunogenic, or it can bypass the p53 deficiency, which has less of a cell death. It can induce an independent apoptosis. Or it was also triggered, that is something that it shares mainly with also high doses of photons, but carbon ions were also shown to trigger, again, independent of the p53 status, a ceramide pathway, which is also related to an increased immunogenicity. Another important feature that is somewhere in between the antigenicity and more on the parts of the adjuvanticity is the type 1 interferon signaling. This is based on a pathway called SIGA-STING pathway, which is triggered, it's normally related to the defense against viruses, bacteria, pathogens, but also for self-DNA from the cell or from extracellular tumor DNA or cells that were phagocyted. Then upon the processing of this material of the cells, we have double-stranded dsDNA in the cytoplasm, which is sensed by SIGAs, which would then activate STING, leading in the end to the release of type 1 interferons. So this is a very important pathway, and has been shown it is very important to attract further immune cells, NK cells, that would lead to the direct cell killing, which also then happens to attract more of dendritic cells, which take up the antigens and would activate the T cells, which again act against the tumor cells. So this is also a very important component. And this has been shown also to happen upon irradiation. So ionizing irradiation can induce the occurrence of double-stranded dsDNA in the cytoplasm, It is not known where exactly it stems from. Could be that it stems from mitochondria, which feature DNA, that it is released by them. It could also be from the nucleus, from the cell nucleus, where the DNA is actually located. So the origin upon irradiation is not exactly known, it's not clear. It can be also both. But radiation does trigger via this mechanism. It does trigger SIGA's sting and leads again to the release of interferon, which is immunogenic. This is some data from GSI, where we could show actually that carbon ions are more efficient in triggering this dsDNA foci, which means that they are potentially also more immunogenic. Along with triggering all these processes, immunogenic cell deaths, adjuvanticity, antigenicity, another important component with the immune system is that you actually irradiate or do not irradiate the immune cells, the potential immune cells that are then taking action in the systemic defense. And this is of really big importance, and it has been shown, for example, that if you take was shown in this paper from 2013, that if you deliver 0.5 gray, so a single fraction 0.5 gray, you already hit 5% of the circulating cells. And in a regimen of 13 fractions, for example, 99% of the pool of blood cells have received low dose, but they have received a certain dose. So what's needed is that you reduce the integral dose, the total dose that you give. You need a high dose range, or you hypofractionate. Those are the things needed to reduce that. And this exactly features with where you can best use hadron therapy for. It has been also shown that particle therapy can better spare immune cells. It's shown here for protons. This is the difference to the baseline. So how much of cell inactivation of lymphopenia I actually got, and this was improved with protons. So there was less of an effect with protons, less lymphopenia, and it has been also shown for carbon ions. This is a study where the blood of patients was compared from photon treatment versus carbon ions, and in the patients treated with carbon ions, the fraction of aberrant lymphocytes, so the lymphocytes featuring chromosomal aberrations, was actually reduced. So you do better spare them, the lymphocytes, the circulating lymphocytes, which are also potential immune cells. You do better spare them with particle therapy. It's not only important to spare the blood cells itself, but also the draining lymph node. The lymph node is often on purpose irradiated to avoid that some metastases that could already be in this lymph node are spared. So you do several times irradiate them, but it's actually considered something that is adverse if you consider the immunogenic effect, because this draining lymph node is very important. This was shown in this study years ago. Injected fluorescent dye, which was then traveling to the lymph node. So what they did was they irradiated either the tumor, only the tumor, or they irradiated both the tumor and the lymph node. And with this, they could show that if you irradiate only the tumor or the tumor and the lymph node, they could show that in the tumor, especially 48 hours after the therapy, you have much more immune cells in the tumor taking action. This was reduced and attenuated when both the tumor and the draining lymph node were irradiated. This translated also in the activity of these cells. So the cytokines and chemokines that are related to a response, to the immune response, were upregulated. But this was attenuated again when also the lymph node was irradiated. It translated also in the survival of these animals. If you see here, when only the tumor was irradiated, the survival was pretty much improved as compared to the controls. But when they irradiated also the lymph node, the survival was very close to the untreated controls. There's also clinical data, of course. Many clinical studies are ongoing, some of which with good outcomes, some of which have bad outcome. It depends on how they are tailored. But this is a very, very famous example of a clinical study of some years ago. I think it was in 2018, where the authors treated patients with non-small cell lung cancer that were resistant to other therapies. They gave to one metastatic lesion hypofractionated scheme of radiation and combined it with immunotherapy, with checkpoint inhibitors, and could show that roughly one third of these patients responded to this therapy actually. And they could also show that this was acknowledged to the immune response, to the immune effect. When you see when they clustered the patients for progressive disease or the responders, especially the ones that responded very good, had an activation of the immune system. And this is interferon, the type 1 interferon signaling, in this case released in the blood, and they could measure that this was activated. The same happened for the expansion of the T cell clones. The cytotoxic T cells that would then inactivate the tumor was much more activated in the responders. When you want to study this in vivo, you normally use these abscopal tumor models where you inject an established tumor cell line in two sides of the mouse, normally in the legs, on one side and contralateral on the other side. You irradiate only one of the tumors, the other one you keep out. You can also give immunotherapy along with, and then you measure the tumor size along the course of the experiment. And you would see whether also the abscopal tumor reacts or not. These models can also spontaneously develop metastases, for example, in the lungs, which you can also assess. These models are somewhat criticized. They are somewhat artificial, of course, because you take an established cell line and you inject it, and then the probability is very high that this tumor kind of reacts. And then it does not need automatically mean that this also will translate in an effect in clinics. And some argue that it is better to have a tumor model that grows naturally. This is absolutely true, but to assess it and to compare it is then a kind of difficult. So that's still the best that we can get so far. Those are some examples of combination in these models of carbon ions with immunotherapy. The authors here compared 5 gray of photons to an isoeffective dose of 3 gray, roughly 3 gray of carbon ions, isoeffective with respect to clonogenic survival. They compared the tumors directly irradiated, where ions were applied, or the out-of-field tumors, which are the abscopal tumors that have not been irradiated. And as you can see, already carbon ions in the abscopal tumor without anything else, at least in one mouse, could respond and could delay this a little bit of growth. But if you combine it then with checkpoint inhibitors, you see that several mice responded. That's shown also here in the survival of the mice, which was in the combination therapies of the checkpoint inhibitors together with irradiation, improved, and is slightly better with carbon ions as compared to the photons. They could also show that in the abscopal tumors and the out-of-field tumors, activation of, in this case, a cluster of T cells and NK cells was happening, so they could show that these were clearly immunogenic effects. In this study, they showed another model the same, so also here, carbon ions in combination with checkpoint inhibitors, and they could show that they do have their effects on the abscopal tumors. They in combination decrease the growth of the abscopal tumor, and could also, they checked also the metastatic nodules in the lung, were also decreasing these. This is a study that you have seen before in the talk by Walter already, in which we compared, in this case, physical isodoses, 10 gray, both cases of photons of carbon ions, and we combined them with checkpoint inhibitors again. So on the primary tumor, the irradiated tumor, of course, the growth was pretty much controlled whenever you give irradiation. In the abscopal tumor, you could see that only the combination therapies would slightly reduce, would actually reduce the tumor growth. What was more striking was, again, the metastasis. And we could see that after carbon ions, especially if you compare it to the checkpoint inhibitors, they had a similar effect. Yeah? So only the carbon ions itself actually led already to a reduction of the lung metastasis, which was not the case for photons. And then in combination with the checkpoint inhibitors, you had a pretty strong effect. There are other ways of combining immunotherapies, other immunotherapies that you can combine. This is an example of Japanese studies where they used intratumoral injection of dendritic cells, which then can also kind of boost, as a kind of a natural boost of the immune system if you inject dendritic cells right in the tumor. And they could show that also that led to an improved survival of the mice. And when they re-challenged the mice with living tumor cells after the treatment, they could see that especially the ones that received both carbon ions and dendritic cell treatment were more resistant to the re-challenge of tumors. Yeah. Okay, I think I have to move a bit faster. So I just want to point out that also there are several things that you have to consider in these models, in these things. One is the sequence in the combination therapy. So when do you give your checkpoint inhibitors relative to the time point of irradiation? In this study was shown that it's better during and after, but not before. You lose a little bit the effect if you give that before. This study here showed kind of similar, so you have to give it either along with, so very close to or after, but definitely you do not give it before. And that was also in this study. And this study, the nice thing of this was that they could show that if you give the immune therapy before the irradiation, before when you do the irradiation, you have given it already before, the outcome is worse. And they could relate this actually to the induced cell death in the activated T cells that were already working on the tumor. You irradiate these T cells, they're more sensitive, and they actually die for apoptosis. It is also important to consider dose and the fractionation scheme. It is not clear in literature. You actually find both. You will see that some say you need a high dose, a single high dose, which is most efficient. You will also find people say no, you need an intermediate dose or a fractionated, hypofractionated, which is more efficient than a single dose. So these are something of ongoing discussion. But in both cases, HITRIplus therapy actually allows for a precise delivery of higher doses, which then actually a single dose or hypofractionated. Which of them is more immunogenic remains to be elucidated. Something else that is, I would say maybe interesting to know is that radiation can also be combined with mRNA vaccines. mRNA vaccines that got very famous in the pandemics, but which were actually tailored for individual therapy against tumors. And this has been also combined with radiation, which is showing some very impressive results, where the tumors after combined therapy of mRNA plus radiation actually several animals responded. In this case, seeing here also an improved survival. That is something very interesting. And in the end, I would just like to show that there are also some clinical trials combining particle therapy and immunotherapy. It's not too many trials, and especially for carbon ion therapy, this is very limited so far. Just 3, to what I know from now, just 3 have been registered, but we will definitely see some more of this very soon. And maybe to conclude, the take-home messages. So I hope I could show you that the immune system plays a pivotal role in the treatment of cancer, especially metastatic cancer. And immunotherapy nowadays is actually established already as an additional pillar in cancer therapy. So the mechanisms by which the immune system is triggered and can interact with radiation are not well understood, or especially not well enough to reliably exploit it in such combined treatment strategies. So a lot of more research is needed. And I hope I could also convince you that HITRIplus therapy has the potential to improve the outcome of such combined treatment therapies. And with this, I would like to conclude. Thank you very much for your attention, and I'm happy to take questions.
Thank you very much for these details, impressive. So please keep your presentation there. There is only one question that we put in the chat. What are delta ALC in slide 30?
This is the absolute lymphocyte count. So the absolute lymphocyte count, and this is delta of this to the baseline. So this shows the changes that you have from the baseline that you see in patients in the blood cell count, and you see that the changes are less with protons. So this means protons are actually more saving. They have less changes, means they have less lymphopenia.
Thank you. Maybe I can take the privilege and ask. I understand that this is a very complicated topic, but are there any recommendations regarding the time interval between the end of the radiotherapy, particularly for carbon therapy, and the delivery of medication for the immunotherapy?
Clinically, not really. So you've seen there were some studies suggesting that you better give it along with it or after it. But the studies are also limited. These are preclinical data being also very limited in terms of you would need a real fraction, a real treatment scheme to better see that. But this is difficult in these models. So there are some ideas on what would be better, like giving it concomitantly or after. But a real recommendation, what you should do, especially with carbon ions, can't be given for now.
Thank you very much. Any other questions? I don't see raised hands. Not in the chat, not in the shared document. So then we thank you